Greetings to you all. In this video lecture, I will be discussing you about cholesterol metabolism. As it is quite visible on the board that the cholesterol is a waxy substance which is found in the blood. It is a fat-like waxy substance, fat-like waxy substance found in the blood. Now it is synthesized by most cells of the body, especially liver and intestinal cells, and it is not synthesized by the plants. We also get cholesterol from certain foodstuff like egg yolk and meat. They are the foodstuff which provide us certain amount of cholesterol. Uh, cholesterol also have got certain uses in our body if it is present in our body in controlled amounts. So this cholesterol is responsible for cell membrane formation. It is responsible for the synthesis of certain hormones. It is responsible for vitamin D synthesis, and it also helps to digest the food, fatty food especially. If we see the structure of this cholesterol, we have a central ring ABCD. It is a central ring. It is a sterol empathic ring with steroid nucleus, and there are 27 carbon in the structure of cholesterol. And the molecular formula, if you see, it is C27 H46 and O. This is the cholesterol formula. And if you'll see this diagram on the right side, it is an animated version of cholesterol plaques which are developed in the blood vessels. And if such plaques develop in the heart vessels, then there are chances of cardiovascular arrest. Now, especially when we talk of the synthesis of cholesterol, how does this cholesterol is synthesized in the body? Now, it is synthesized by most cell of the human body. That means almost all the cells have got ability to synthesize cholesterol. But especially this cholesterol is synthesized in the cells of liver and intestinal cells. They are the two sites, two organs where this cholesterol is especially synthesized. But it has got all the cells of the body have got ability to synthesize cholesterol in some amount. Now, this molecule acetyl coenzyme A, it is a precursor for all 27 carbon of the cholesterol. That means total structure of this cholesterol is synthesized from in starting material, that is acetyl coenzyme A. This cholesterol synthesis completes in 5 steps, in which the first 2 steps occurs in the cytoplasm and last 3 steps occurs in the endoplasmic reticulum. Now we will talk for the synthesis of cholesterol step by step. So this is the step 1. In this step 1, 2 molecules of acetyl coenzyme A, they actually give rise to mevalonate, and this is the condensation of 2 molecules of acetyl coenzyme A. Now in this first step, it is called as principal regulatory step because the enzyme HMG reductase here, which converts 3-hydroxy-3-methyl-butyryl coenzyme A into mevalonate. Let us see this point in this cycle. So first of all, in the step 1, the 2 molecules of acetyl coenzyme A get converted into acetoacetyl coenzyme A. Here coenzyme ASH is removed out and the enzyme is thiolase. This is the first part of the first step, and the second part of the first step includes the conversion of acetoacetyl coenzyme A into 3-hydroxy-3-methyl butyryl coenzyme A, which is in short written as HMG coenzyme A. Here the enzyme is HMG coenzyme A synthetase. Now this enzyme is regulated by the high concentration of cholesterol. If there is high concentration of cholesterol, then this enzyme will be inhibited and the first step is blocked. So this is the feedback regulation. And also by use of certain drugs like statins, this enzyme can be blocked. So whenever there is high concentration of cholesterol in the body, then the doctors recommend statin drugs which blocks this first enzyme, and that's why it is regarded as principal regulatory step, the first step. Regulation is taking place in the first step only because of this enzyme, because this first enzyme is inhibited by the high concentration of cholesterol and also by certain drugs like statins. They are responsible. Now further, this 3-hydroxy methyl butyryl coenzyme A, it is converted into mevalonate. The enzyme is HMG coenzyme A reductase. Here, two molecules of NADPH2 is converted into two molecules of NAD, and coenzyme ASH is removed out here. The mevalonate get converted into isoprene units, isoperinoid units. This is the step 2. Here, 3 molecules of ATP along with magnesium ions are used, and the CO2 is released out. Here, phosphorylation and decarboxylation of intermediate substances takes place, and that's why the final step is mevalonate get converted into isoperinoid. In the step 3, we have got the formation of squalene. Squalene is synthesized from isoperinoid units with the help of squalene synthetase, and here 6 isoperinoid units condense with the help of this NADPH2, which get converted into NADP, and 2 molecules of phosphates are removed out here. Finally, the squalene is formed. And then there is a structural change of squalene. The squalene, which is initially a linear structure, becomes circular, and then this is known as cyclization. So there occurs cyclization of this squalene to form linosteroid, and the enzyme is cyclane here. Here also NADPH2 is converted into NADP and oxygen is used. In the last step, step 5, the lanosterol, which is a steroid nucleus, is modified to form cholesterol with the help of NADPH2, and the intermediate products includes 19 steps here. But actually, it is the formation of cholesterol from lanosterol. This is how the final synthesis of cholesterol occurs in liver cell, intestinal cell, and some other cells of the body. So these are the write-ups of step 2, step 3, step 4, and step 5. So you can read these steps later on, and this is all about the cholesterol synthesis and introduction of cholesterol. About the other parameters of cholesterol, like regulation of cholesterol or excretion of cholesterol, I will be dealing in a separate video. Thank you very much.